So, let me first start about some of the basic concepts of proteomics, especially an overview of proteomic technologies from discovery to function. So, let us first look at the taxonomy of omics. I am sure you have been listening variety of terminologies linked to omics field. Omics field aims to look at a given system in its totality. For example, all the genes or DNA of a given system can be studied under genomics, all the RNA or transcripts under transcriptomics, all the proteins under proteomics, all metabolites as metabolomics. Let us say if the context was to look at all the possible transcripts, genes or proteins, then you say that we are going to study the global proteome or global genome analysis. But if the concept is linked to a specific cell or an organ, then that is cellular or organelle proteomics. So, in this way I hope that you are clear about layers of information and different type of omic terminologies. All this started from the success of the human genome projects and the success of genomic technologies, especially in the 1990s. That time various genome projects were in progress, and especially 2001 and 2003, during that time the draft human genome maps were published. It was one of the major accomplishments of first time knowing about all the possible human genes. While this was one of the major breakthroughs which has happened in front of many of us, a major milestones in biology, but when people looked at variety of model organisms, especially fruit fly, which is Drosophila melanogaster, roundworm, C. elegans, Thailcrass Arabidopsis thaliana, human Homo sapiens, variety of model organism show the number of genes are not very different in different systems. The numbers may vary around 17,000 to 20,000. So, what makes different systems so different morphologically and physiologically? So, eventually the focus shifted from the genome moving on to the protein level investigation or proteome. Let us look at some of the basic information, basic biology. From the genomic DNA, after transcription the RNAs are formed, and in the process of splicing you can see that the exons and introns are there, and when introns are out the functional mature mRNA is being formed with combination of various exons. Now, during this process from pre-mRNA to mRNA or the mature mRNA formation, variety of combinations can happen, different exons can come into different combinations to give rise to different type of mature mRNA, which may eventually result into different protein forms. So, from same gene different transcripts and different proteins could be produced. And further, now this information could be much more complicated because each protein may also get modified based on the attachment of phosphate residue, methyl residue, glucose or the sugar molecules, hydroxyl residues. Many of these modifications known as post translational modifications are very relevant for the functional information of the protein. So, looking at this information, now let us look at this cartoon where we can see the inner sphere, that is the genome, then the transcripts are being formed, the blue circle shows the transcriptome, and then eventually the proteins are formed and gets modified, that is the proteome. So, study of proteins and their properties to provide a broad integrated view of cellular processes is known as proteome, and the field which aims to study that is known as proteomics. I would like to highlight that after genome reference maps which are published, the eventual goal of the entire community was to look at possibility of all the proteins and trying to obtain the evidence that these proteins do exist in human with various type of experimental approaches like mass spectrometry and tissue based microarrays. All those efforts took a lot of time, but eventually 2014 and 15 the first draft human proteome map was published, which tried to capture and provide evidence that proteins do exist for all the genes which were already decoded, and then almost 17,500 proteins were the evidence for those were provided. in 2 different seminal papers based on the mass spectrometry in 2014 and then eventually based on the tissue based map of the proteome in 2015. So, these are again the major milestone accomplishment. After the draft human genome maps, we now have the draft human proteome maps available. So, broadly if we want to study the proteomics or the field of proteomics, you can think about there are 2 major objectives. One is to look at the new discoveries or new kind of targets in context of a given disease or given stress or given comparison which you want to make, or you want to understand the function of the proteins. So, one could term as the discovery or abundance based proteomics, another can be known as function based proteomics. So, discovery based proteomics aims to look at the new protein molecules which could be showing change in the abundance because of the disease or stress condition or drug treatment, and what are their identification and what is their quantification. So, these information could be obtained under discovery or abundance based proteomics. Then one could look at function based proteomics, where aim is to identify the function of unknown proteins or the new proteins which you have discovered from the discovery set. So, where the proteins are expressed, the assays to try to understand the function of these proteins are the major goal of functional proteomics. So, if you look at the technologies which could be used for discovery proteomics, essentially various type of gel based technologies like 2D gels and dyes and mass spectrometry were quite handy to use the discovery based workflows. And then how to look at the functional information was based on the protein microarrays and SPR, surface plasmon resonance based technologies, which provided more information at the functional level. So, much more gel free kind of approaches, more high throughput approaches were used for the functional studies. So, one could say that there are various facets of proteomics, starting from the beginning of the proteomics field, which is started with gel based proteomics or the gel free proteomics, which is predominantly based on the mass spectrometry and various label free biosensors, or functional proteomics where you want to study the function of the given protein, or targeted proteomics where you want to target a specific protein or peptide sequence and do their validation in the high throughput manner. I will give a very brief overview of each of these domains. Of course, there are separate courses which I have offered in the past, where you can get much more detail about each of one of these technologies and the full modules on those, but let kind of capture very briefly the various technologies available for studying proteome. So, in this slide gel based proteomics comes very first, because all of you have studied at least some level of gels, LDS page gel or even 2-dimensional gel electrophoresis, or the advanced form of gels which is known as dyes or difference in gel electrophoresis. So, the study of looking at the proteome or separating proteins on the gel have been from very long time. Electrophoresis field has existed from 1950s, and especially the 2D gel came into the light in 1975 when Close and Farrell they first time showed that one could separate proteins based on their 2 properties of molecular weight and isoelectric point. So, this is the cartoon diagram showing you the concept of 2-dimensional electrophoresis, where the first dimension protein separation can be done on the IPG strip or immobilized pH gradient strip in the isoelectric focusing field based on their PI values. When pH equals PI, there is no net charge, then protein will stop migrating in the electric field, and this is how you can separate the proteins based on their PI values based on either 3 to 10 IPG strip or 4 to 7 IPG strip. You can choose as per the relevance. Once you have done the first dimension separation, then put the same strip on the SDS page gel unit, and now you can separate proteins on the second dimension based on their molecular weight. So, then eventually you generate an image which is shown in this particular spot, where you can obtain information for each protein at their molecular weight as well as isoelectric point level. Now, let us say that your goal was to do the differential proteomics or abundance based proteomics to look at the new targets in context of a given condition A and condition B comparison. So, you have done the protein extraction, solubilization of the proteins, and then separated them based on their isoelectric point in the first dimension. After reduction and alkylation, proteins are separated on the SDS page gel in the second dimension. You can now separate those 2D gels, you can now stain those for visualization purpose using either Kumasi stain or silver stain. Now, these two gel patterns are emerged from condition A and condition B. After analysis, you can see that some spots are differentially expressed, and if those are reproducible, you can excise or cut the spots and then do the protein identification using mass spectrometry. While this is all very straightforward, very nice technology, but because of the reproducibility issue, because of the running artifacts, and because of the the low throughput, the number of protein coverage issues, a new technology was much more easy to accept, that was dyes technology, which is difference in gel electrophoresis, especially for the quantitative proteomics. So, in this particular technology, let us say you have two samples for condition A and condition B, which you want to compare. You can now label them from Psi 3 and Psi 5 dyes. These are fluorescence dyes from the cyanine, and then in addition to these two dyes, you are also adding making a mixture of the both the samples from sample 1 and sample 2 as a reference pool, and then use that reference pool to label with Psi 2 dyes, mix all the 3 samples together, run on one gel, and then you are going to obtain the 3 images after scanning for Psi 2, Psi 3 and Psi 5. So, in this slide, now you got control and treatment, Psi 3 and Psi 5 images, and Psi 2 as the reference pool. Let us say if you have to run 3 biological replicates, in all the 3 biological replicate, Psi 2 image will remain the same, because you have already made the pool of that, and the control and treatment images will change. So, this became much more reproducible, much more quantitative, and if we now want to summarize, I have just shown you data from the lab, where now you can see after comparison, these are the 3D views of the spots from the dye gel. The bottom panel is for the control spots and the top panel for the disease spots. So, looks like something is very interesting 3-dimensional view, like showing higher expression of this protein in a given disease context. So, if we want to summarize the overall what we learned from the gel based proteomics, that gel based proteomics are very handy, very simple, easy to do technology, but because of the reproducibility issues and limitations of gel to gel variability, the focus shifted more towards the quantitative proteomics based on the dyes. Dyes is a multiplexing technology which offers much more sensitive and much more reproducible way of doing gel based proteomics. However, the major challenge of both the 2D gel or dye gel remains that after looking at the significant protein spots, you want to identify them using mass spectrometer. But from the gel, when you excise a given spot, the protein amount present in that spot may be too little, too low for you to reproducibly capture that information and then get any peptide identification. So, even now you see this particular 3D view spot looks pretty nice and biologically very relevant, but what this protein is, we have no idea, no information. So, can we now identify the protein using mass spectrometer, and if we can do that, then only we will have any sort of biological information. But if mass spectrometer fails to identify this information because there is not sufficient peptides available, then I think our all effort have failed. So, therefore, gel based proteomics is great, it can do many things, but still the success of identifying the protein of relevance could be only 50 or 60%, because you cannot identify all the protein spots. So, a lot of focus shifted in the overall proteomics field from the gels and moving to the gel free proteomics, especially based on the mass spectrometry. So, let us now shift to the next module, especially looking at mass spectrometry based proteomics. I must say this particular technology has almost influenced very positively the entire field of proteomics and even metabolomics, and as a result you will see a lot of developments happening in this area. I will probably give a separate lecture in my next lecture just focused on the MS based proteomics, but let us kind of get very brief overview of mass spectrometry based proteomics. The mass spectrometers, they are aiming to provide you information based on the mass to charge ratios of these particular your peptides to give you identification and quantification, and to achieve that you are trying to separate these ions in the electric and magnetic field. So, definition is mentioned here that this is a technique for protein identification and analysis by the production of charged molecular species and their separation by magnetic and electric field based on their mass and charge ratio. This image is shown here is for shotgun proteomics. You do not want to use the gels, directly solubilize the protein, do the reduction, alkylation, followed by treat with enzyme like trypsin for digestion. Now, all the proteins will convert to the peptide form. These peptides can now be cleaned up for the salt and other debris. You remove those artifacts, and now the clean peptides could be directly subjected and injected to the inside the mass spectrometer for doing MS analysis or MSMS analysis. In this area, you will also use the chromatography, like especially strong cation exchange or reverse phase for peptide separation, and then you will use ionization source like electrospray ionization or MALDI to ionize these peptides into the gaseous ionized forms, then separate them based on their various M by Z properties in the mass analyzers. There are different type of mass analyzers available, could be quadrupole, time of flight, ion trap, orbitrap, many type of configurations are available, and then one could use MSMS to generate the peptide spectrum and then use databases for peptide search, and eventually you will get the hit for the protein identification. In just nutshell, of course, we will touch upon some of this concept in much more detail in the next lecture, but I just trying to give you the overall field of proteomics, especially gel based we covered, and now I am trying to give you the feel for the mass spec based proteomics, especially shotgun proteomics. So, if you want to do quantitative proteomics using mass spectrometer the way we talked for gel based, where we have used the concept of dye-etch technology, here one of the successful example is I-track based quantitative proteomics. I-track is isobaric tagging for the relative and absolute quantification, and here again you can take now 4 samples or 8 samples, or even a similar technology like TMT or tandem mass tag have come forward, where you can study even 10-plex studies. These are isobaric tags, so it means you are not changing over addition of the mass on any of the 4 condition or 8 condition. So, in this way we have this I-track label 114, 115, 116, 117, report the balancer region is 31, 30, 29 to 18, overall mass added is 145, which is all same in all the 4 conditions. You have labeled the peptide of 4 different conditions, mix them all together and do MSMS analysis. This cartoon shows you that you have 4 conditions, you have done protein extraction followed by digestion from those, label the samples using I-track labels, combine them, do the some sort of chromatographic purification if required, and then followed by LC-MS analysis. At the MS level, you can see these reporter ions could be visualized 14, 115, 116, 117, and this information could be used for the quantification of peptides. So, this is the in a very nutshell, very brief about using mass spectrometer directly for the quantitation. You are not using any gels in this approach. Next technology which is coming forward is targeted proteomics, which is also based on the mass spectrometry revolution, where aim is to look at peptides directly using triple quadrupole based mass spectrometers and do their validation. So, one approach which has come forward very promising is known as SRM, selected reaction monitoring, or MRM, multiple reaction monitoring. And in these technology approaches, these assays, you will see there are triple quadrupole mass specs being used. For example, Q1, which is a mass filter, here you are going to select a given peptide. So, you already identified the peptide sequences of interest from discovery workflow. Now, you want to validate a specific peptide. So, you are selecting those specific peptides now, let us say shown in different colors of red, blue and black. For the red one, now you are doing the fragmentation in the collision cell and then further analyzing its product ions. So, you are looking at the precursor and product ion pair, which is referred as the transition, and then you are monitoring at least 3 transition of a given peptide, something shown in these cartoons, and you are looking at least 3 peptides for a given protein to be very confident about your quantification. So, now using this information that for a given peptide you have measured at least 3 transitions, and then for a given protein you have taken at least 3 peptides, now you can validate peptides directly using mass spectrometer, and of course, you will have much more detail eventually in the in the lectures as well as some of the demo sessions where you will be introduced to the concepts of promising software like Skyline, how to analyze the targeted data and how to conduct these experiments using targeted proteomics, triple quadrupole mass spec in the lab setting. We will also have a demo session on that. One could utilize these information for the actual clinical work. For example, now we identified some specific targets for one of the studies in the brain tumor, and now we are only looking at that target across large number of patients. These are the patients suffering from various grades of gliomas, grade 2, grade 3 and grade 4. This particular protein L-lactate dehydrogenase, we are trying to measure only that peptide across all of these patients, and we see there are much more abundance of this peptide in grade 4, and something similar result we had also seen in the discovery workflow when we use the iTrack based work. So, you can see that there are different technologies which have complementary nature, which could be utilized to complement the information, and rather than now generating the antibodies separately for doing validation, you can just use the mass spectrometer even for the validation. And of course, once these validation looks very promising on large number of samples, then you can make synthetic peptide which are heavily labeled and then try to do the more accurate quantification. Once even you have passed that kind of roadblock, and you see that now there is a detectable amount, very reproducible accurate quantification of a given peptide from the clinical samples, then you can take it forward next level. You probably now want to develop a clinical assay directly using mass spectrometer, or you can raise antibodies or some other kind of biochemical assays which could be utilized for doing the much simpler based assays like ELISA or Western blot for the clinical utility. So, this can be a path and the workflow for taking your the project of interest from discovery to the validation, and followed by taking to the clinical translation work. So, I know I am kind of going fast, I am trying to cover variety of fields in a very nutshell. Each one of these require a lot more detail for studying about the details of each technology, but let us kind of capture the at least the breadth of the entire field of proteomics, and especially we started from gel based, move to mass spectrometer, and now let us shift slightly towards the interactomics and functional proteomics technologies. So, variety of technologies are being developed in this area of interactomics, where aim is to look at how a protein interacts with another protein, another biomolecule or a drug molecule, and how to identify their function, or at least get a glimpse of what could be possible function of an unknown protein. In this light, traditional approaches like yeast-2 hybrid or even affinity chromatography, immunoprecipitation were heavily used. More recently, we have started using protein microarrays and label-free technologies. This is a complex slide which shows you variety of platforms available for doing protein microarray based work. Let us start understanding it in the small pieces. If you have printed antibodies on the glass slides, that is, let us say these three cartoons, which is known as abundance based proteomics. So, you are trying to measure the abundance of a given protein for which you already have an antibody, and then the way you can develop your assay, it can be direct labeling, it could be sandwich assay, or it can be reverse phase protein arrays. In reverse phase protein arrays, you would like to take the tissue lysate or cell lysate and then probing for a given antibody to measure its abundance. So, this is known as abundance based proteomics, where aim is to measure the abundance of biomolecules by analyte specific reagents. Or you can also look at the function based protein microarray approaches, where aim is to study the biochemical properties or the protein interactions, for which either you have purified a protein and printed that on the chip, or you know the specific peptide sequences and you have tried to use those information for the printing on the chip, or you can just take the cDNA directly. For you have not purified a protein, but directly you are taking the DNA or the cDNA, print that on the chip and use in vitro transcription translation mix on top of that, and then try to synthesize protein on the chip. This field is known as cell free expression microarrays or cell free based proteomics. So, the technologies like nucleic acid programmable protein arrays or NAPA or multiple spotting techniques are promising approaches to do these cell free expression based microarrays, but all of these four which I have showed are possible ways to look at the function of the proteins. So, in some way that there are a variety of platforms available for doing the proteomics, especially even the interactomics using microarray based workflows. Additionally, another technology, if you are now aiming to look at more quantitative information for a biomolecular interactions, then you do not want to label the proteins with any given fluorescent or fluorophore. So, here your aim is to look at the biomolecules in its native state, and you are looking at in the label free manner. Surface plasmon resonance or SPR is one of the promising technology in this manner, which measures the change in the percentage reflectivity as a result of the two biomolecules comes into the binding. you will see the change in the percentage of reflectivity or in the reflective index of the medium will change, and therefore, you can see the binding is happening or still very weak binding is happening, and that is measured under the sensorogram, which I will show in the next slide. But here this cartoon what it shows that you have a gold slide, you have a light source and you have immobilized some antibodies of interest. For example, you are passing your proteins of interest to measure that there is binding happening for this protein or not, and then if there is a binding, then you will see the change in the percentage reflectivity, and followed by you will monitor the sensorogram. So, if you now look at what you obtain from this kind of experiment, initially you have a straight baseline, you are now measuring the sensorogram, which is response unit versus time scale. The baseline is very straight, and after a binding starts happening on the chip, you will see the on rate, and then you keep flowing the buffer, so that the molecules can now dissociate, and that will come under the dissociation phase or the off rate, and then you want to utilize the same chip for further experiment, and that could be studied after regeneration, when now you can use some of the mild acids to strip off your binding. So, this is the change in the SPR signal versus time is known as SPR sensorogram, shows you the binding activity and provides information for the on rate, off rate or the Kd values of a given interaction. A new advancement in this area is SPR imaging, where now the aim is to look at something like combining microarray based concept to the SPR based concept. You want to study the molecular interaction in a high throughput manner like microarray, but also want to get their Kd values and the quantitative information in the high throughput manner, and that is where SPR imaging is one of the promising approach. Additionally, one could look at identification of new interactors. Let us say you have an antibody which you have immobilized on the gold slide, you are passing a cell lysate or tissue lysate, and you are expecting to bind some of the unknown proteins on the antibody. So, therefore, you want to now identify which are the potential interactors which are binding to this antibody of interest. So, can you now identify those interactors, and that is where you need to bring in the SPRMS, a new technology approach. So, can you now strip off this particular bound molecule or the analyte which is bound on the gold chip, and then generate sufficient of those peptides, so that now you can run on the mass spectrometer. We have tried to optimize some of these technologies, and after multiple run cycle you can now generate sufficient amount of peptide, which has given us detectable amount of peptide in the mass spectrometer, and you can identify these interactors or the proteins of interest. So, finally, what we are doing, we are trying to build the layers of protein information using variety of proteomic technologies, where proteins are localized, how they interact, which are the substrate for these assays, and variety of ways we are trying to understand the protein function, which is very complex. So, in this slide I would like to conclude that there are many promising proteomic technologies are in development. You need to be very cautious and clear which technology platforms you are going to use for your addressing a biological question. All of these proteomic technologies have made huge revolution of understanding various biological system, but depending on your need, you can choose sometime a gel based proteomic technology or directly mass spectrometers, or use microarrays or SPR or many other technologies available to address the right biological question. So, depending on what is your question, what you want to obtain the answer, you can choose the right type of complementary proteomic approach. I hope it kind of gives you the the good foundation and at least the overview of the field of how various type of proteomic technologies could be used for studying any biological system. Today I am going to talk to you about some of our ongoing research work to give you the flavor of how proteomic technologies could be used for different type of applications, especially more focus on clinical proteomics. This in some way is going to summarize that different type of proteomic technologies could be used to address different biological questions. As we go along after this lecture, there will be much more focused discussions about mass spectrometry based proteomics, different softwares and tools involved by various experts of the field who are going to talk about much more detail about how to use various tools for data analysis. So, this is the third lecture of giving you the basics and overview. After this, there will be much more focused discussions and the hands on directly from the workshop. So, just talking about various type of ongoing projects, especially in my laboratory at IIT Bombay, we look at some of the technology platform or the assay development as one of the areas of interest. A major group of the lab works on the infectious disease, especially malaria and dengue, and try to look at different type of either diagnostic biomarkers or looking at the prognosis, how a disease transforms from the non-severe to the severe form, and how using proteomics one could try to understand that. Additionally, a major focus in the lab is on the brain tumor, especially cancer biomarkers in collaboration from Patna Memorial Hospital. And then we have various ongoing projects from different collaborators on other model organisms. For this brief presentation, I will focus mainly on two different disease projects, one on the infectious disease and other on the cancer projects, and try to give you a broad understanding about how proteomic technologies could be used to study different type of clinical problems. Let us first talk about infectious disease or malaria proteomics project. Malaria, I am sure all of you are familiar. As the monsoon season starts, people starts getting different type of mosquito bite and get affected from either falciparum malaria, vivax malaria, or sometimes dengue fever, chikungunya, variety of fever you can term, and it becomes very difficult for the clinicians and the doctors to really make accurate detection of what kind of the organism has caused this particular type of fever. And if you get the right diagnosis, only then you are able to get the right type of treatment, right. So, the interestingly looking at malaria, the trend for malaria pathogens have slightly changed over the time period, especially last couple of years when we have started investigating this problem in India. We found that falciparum malaria have reduced, whereas vivax malaria has quite increased now, and you can see the trends in different regions of India, where it shows that now vivax malaria is pretty much on rise. While earlier this particular vivax malaria was thought to be very benign and not causing much of the problems for malaria, but now looks like it is one of the major culprit which is causing lot of malaria problems in whole India, and of course many parts of the world as well. So, however, our major understanding of malaria predominantly depends on the falciparum malaria. Lot of research has happened in that particular pathogen, but when it comes to vivax, it has been very limited because of various reasons of our inability to culture the vivax in the culture system, which is not the case for falciparum, and also the vivax even at a very low parasitemia level, it could cause even the severe effects, which is not the case again for the falciparum. So, current modalities of diagnosis of malaria parasites based on the microscopy, or it could be rapid diagnostic test, or it could be PCR. While microscopy is gold standard, when the pathologist they look at the the parasites in the microscope, and if these are the trained eyes who can look at the parasites well, I think that is very accurate way of detecting parasite. RDTs are very quick, these are based on some antigens, when you can do some strip based test quickly to tell if a pathogen is coming from the falciparum or vivax or mixed infection, and one could try to get that information, or the PCR could be utilized to be very accurate that which kind of pathogen is causing this particular febrile response. However, each of these methods have their own pros and cons, especially RDTs have lot of false positives. PCR can be done only in the specialized labs, so it needs lot of further testing. And overall limitation for the whole field has been that most of our information is based on falciparum, not vivax. Therefore, everything is we try to put together based on the falciparum and say is that falciparum positive or negative, and accordingly whether this could be vivax. So, there is still need to look at specific antigens for vivax if we really want to investigate this problem. So, in this slide one could look at different type of proteomic approaches, different type of biospecimen to try to investigate this problem. Let us say one question we will try to address: how does the proteome modulates in response to Plasmodium falciparum or vivax infection? To do that, we looked at the patient's blood sample and try to look at the plasma as well as the erythrocytes, the plasma proteome to look at the host responses, and the erythrocyte for looking at the parasite proteins to look at what are the parasite proteins which one could detect directly from the clinical isolates. So, both of these strategies in some way are complementary to give us very vast information about the parasite proteome and the host proteome to find out what is happening in response to this malaria in these patients. So, one of the goal of the project was to look at, can we identify some specific biomarkers which could be very unique for the vivax malaria. A lot of work has happened in the lab, but just in a nutshell, in one of the study where over 200 patients were enrolled, we looked at parasite proteome. We also look at which of the parasite proteins are secreted in the plasma, because those are circulating in the blood and those could be much more relevant biomarkers for our study. And therefore, some of these biomarkers we try to now do the bioinformatic analysis and see are they only uniquely present in vivax or whether they are also present in the falciparum. And interestingly, many of at least 5 candidates looks very promising only present in the vivax, which could be next generation diagnostic biomarkers. Now, there is a need to take these leads forward to clone, purify these proteins and then see with the possibilities of having the RDTs based on these protein biomarkers. Additionally, we looked at now a protein microarray based platform to know which antigens are markers of exposure in malaria. To look at this, some of this study can be foundation for doing the population based studies, where one could ask various questions. What is the antibody breadth for a population based survey? And in this case, we looked at from the Goa population in collaboration from University of Washington, Professor Pradeep Rathod's lab, variety of patients affected from malaria, both falciparum and vivax were tried to screen, and on the chip we had only the antigens from falciparum or vivax parasites. So, now the patients serum samples are probed on the chip and we are looking at antibody breadth of the patients suffering from falciparum or malaria. Also, we try to look at the surveillance kind of responses based on the antigens, which is shown in this particular the middle segment here, where you can see even based on the age groups one could try to see different type of responses. And in the left side you can see based on the falciparum or vivax, the patterns are different for the seroreactive antigens which are recognized by these patients. And finally, if you look at the right panel, we are looking at also the severe and non-severe responses in these patients, and you can see a very different pattern from the non-severe versus the severe patients. So, again this could be one of the interesting approach of looking at the antibody based responses using protein microarrays. Again, to address what are the mechanism of vivax causing the severity, for which we do not have much information, and some of these study I am just talking to you about partially published and some are leads are still unpublished, where goal was to look at if a patient comes to a doctor and they are having the mild fever, whether one could try to look at possibility of them transforming to the severe form and what kind of proteins and metabolites could be indicative of the severity. So, in this slide we did a quantitative mass spectrometry based workflow, where the healthy individuals, non-severe vivax malaria patients, severe vivax malaria patients were screened, along with them we use a reference pool here. So, all of this a typical eye tract based mass spectrometry study which we talked in the previous lectures was employed, and what it revealed that there are set of proteins which are very unique to non-severe or the severe vivax patients, looking at both the proteomics approach and even after metabolomics also we found that there are some very unique metabolites which are secreted here. Some of the proteins from the acute phase pathway and the cytoskeletal proteins are shown on the screen here, when a protein like serum amyloid A shows from the healthy individuals to the non-severe to the severe patients kind of rise of the change in the abundance of these proteins. Another protein like haptoglobin shows the reduction overall, the concentration reduced from the non-severe to the severe patients, and that is more logical because the haptoglobin and hemoglobin they form the complex, and therefore you will see the reduction of this particular protein in the severe vivax patients. Some of the cytoskeletal proteins like titin and vitronectin, they showed the increased response as the disease progresses from the non-severe to the severe type, which is indicative of some of the muscle protein and cytoskeletal protein being secreted from the muscles to the blood stream, and that shows now the patients could be modulated from the non-severe to the severe type. We try to look at various metabolites shown in the right side, and then the red dot shows you the pattern of healthy individual, and this part shows the non-severe and the last part shows the severe patterns. Even by looking at the metabolite profile, one could see at least there are some metabolites, especially various amino acids, they show the change in response to the non-severe and severe type of malaria. So, based on this we are trying to now capture which are all changes one could utilize from the omics technologies and try to look at what is the mechanistic insight of vivax malaria severity. We have tried to put together a lot of information from proteomics data and metabolomics data, able not to give you a lot of detail right now for the mechanism, but I am trying to emphasize that how these tools and technology what we are talking can give you the new insights which could probably help you to understand the mechanism of a given disease. So, in this context very first time we are now able to put together various pieces and trying to see that in which way the various changes which are happening which are reflecting from the addition molecule to the inflammation factors, how they are with the change of various type of amino acids and change in the haptoglobin level leading to the oxidative stress or apoptosis, followed by it is influencing the vascular leakage and coagulation pathways, which is eventually causing variety of severe infection which we can see. So, the activation of oxidative stress as well as the the cytoskeletal regulatory molecules could be the one of the major mechanism which is contributing toward the severe vivax malaria. Of course, this is very complex slide which needs lot more time, but I am not going in detail. Here idea is only to give you the the glimpse of what you can try to understand from the proteomics and other omic technologies. Additionally, we are trying to also address more focused questions, like in case of falciparum malaria there are various type of severe infections happens, like patient could be suffering from the cerebral malaria or renal failure or variety of other complications. All these could be termed as SFM or severe falciparum malaria, and question was could we look at proteomic based alteration in the severe falciparum malaria based complications. In this slide we selected the patient from cerebral malaria, severe anemia, other type of severe falciparum type. When we do these kind of proteomic studies or clinical studies, choosing the right control becomes very crucial and of course challenging as well. So, in this case for the anemic population we choose as a control for severe anemia. We also choose various type of non-severe falciparum patients. We got some meningitis patient for the control as a cerebral malaria. So, you need to have the right type of controls to compare the various type of disease complications. Now, after doing lot of proteomics workflow, this heat map is shown here, which shows that one could actually segregate severe and non-severe falciparum based on some of the proteins shown on the right hand side. And even within each type of severe infection, like you can see cerebral malaria, severe anemia, other type of severe infections, their trends for various proteins are quite different, and therefore this information could be helpful to get a glimpse of what type of severe infections these patients might be undergoing if they are affected from the falciparum malaria. Additionally, we are also interested to look at are there some parasite proteins secreted in the serum or plasma of these individuals, and whether those could be used as the next generation diagnostic biomarkers, because some of the existing biomarker like PFHRP2, while it has been a good lead for the RDTs, but there are many populations where now it is being shown that there are some mutations happening and this may not be the best diagnostic biomarker. So, there is definitely need to have the alternative next generation diagnostic biomarkers even for falciparum malaria. And in this light various protein which we try to identify, some of those are already same protein what are available in the existing RDTs, but additionally we are also able to find some new proteins which are secreted from the parasite in the host bloodstream or the plasma, and one of those is serine repeat antigen 4 protein, which looks promising again and could be taken forward as the potential biomarker candidate. So, in the nutshell of the malaria project, we now have various leads, various protein targets both from the parasite as well as from the host, and our aim is to look at what happens to the given protein, like let us say alpha 1 anti-chymotrypsin or alpha 2 HS glycoprotein or complement C3 across different type of complication, from the non-severe falciparum to the non-severe vivax to the dengue fever and severe type of falciparum and vivax. So, what happened to the same protein across multiple infection, and if there is a real signal coming out of in a specific type of infection, you will see a different trend. Now, some of this information we are trying to take forward to develop some of the the possible kits or the assays for the better diagnosis and prognosis of the patients. So, in general the conclusion for the first part of malaria proteomics is we are looking at various approaches of serum proteomics and metabolomic analysis to look for the prognostic biomarkers and understand the host responses. We are also looking at various type of parasite proteins in the serum to look at the potential biomarkers for the diagnosis. We have also investigated the non-severe to severe comparison to try to look at the severity of different complications both in the falciparum and Vivax, and actually nothing was available information for the Vivax, and we have also done much more in depth investigation of the different type of severe falciparum complications. And an offshoot of the overall project is also looking at the antibody breadth and looking at humoral responses of using the protein microarray based workflow to look at various type of markers of exposure of malaria. So, this is where you can see to investigate one clinical problem you can utilize different type of technologies from gel based to mass spectrometry to microarrays and to SPR even to test out the protein drug interactions to then try to understand comprehensively a given problem or a given system. So, let us now shift gear from infectious disease, moving on to one of the clinical problem of a great relevance of the cancer, especially the brain tumors, which is very deadly tumor which affects a lot of patients who do not survive after very long if they are diagnosed with the brain tumors. Depending on the location of the in which region the tumor is, the brain tumor could be termed as the gliomas if they are deriving from the glial cell, or meningioma if they derive from the meninges, or medulloblastoma if they are in the pediatric tumors. So, to do this kind of project we collaborated from doctors from Tata Memorial Hospital as well as ACTRAC in Mumbai, and we try to utilize various type of workflows and approaches how to use proteomics to investigate these problems. But let us kind of give you a brief of gliomas and meningiomas, which are very challenging brain tumors. Most of the these brain tumors are very heterogeneous, as well as if you look at the available information from WHO, that is more based on the cell morphology and some of the immunohistochemistry based biomarkers. There has been extensive genomics which has happened in this especially the glioblastoma multiforme brain tumor, but not much of the overall proteomics or the proteogenomics investigation has happened so far for various type of the low grade gliomas and meningiomas and other type of brain tumors. So, on one hand we definitely need good repositories or for the biospecimen to do these kind of research. Additionally, there are a lot of challenges of the heterogeneity of these tumors which we have described in some of these review articles, which show the challenges of doing this kind of research, because you need large number of samples, and even if you have large number of samples, the patients will be affected from the variety of issues, and therefore so much heterogeneity is there even from the same patient. So, to investigate that you need of course complementary approaches, you need large number of samples, you need very robust data analysis workflow, and together only you can try to obtain some information. So, let us say how we can use the proteomics workflow to address some of these problems. So, you can get variety of sample type either from tissue, serum or the cerebrospinal fluid. Now, use this sample type biological specimen to do discovery either using mass spectrometry based workflow or protein microarray based workflow, and after identifying the potential candidate targets, then one could go to validate the biomarker using targeted proteomics based workflow. So, some of the technology which we talked in my first lecture, let us try to see how we can put them together and use them in this kind of clinical problem. So, we wanted to ask a question whether this kind of proteomics investigation which we are trying to address for a brain tumor could identify some of the key networks and potential targets for different grades of meningioma brain tumors. So, we are now looking at meningioma, and then we are using 3 different type of complementary approaches. One is global proteomics, where aim is to look at all possible proteins and use their quantitation using either iTrack or TMT based workflow, or use the global proteomics workflow using the label free analysis, which is LFQ label free quantification, or DIA data independent acquisition, or we do the phosphoproteome analysis just to look at the enriched phosphopeptide residues available from these peptides. To do this, let us say from the same patients, the brain tumor sample when it comes, you can do the protein extraction and split those particular peptides after digestion into multiple tubes, and now each tube containing peptides could be utilized for either iTrack or TMT workflow or LFQ or DIA workflow, or you can use for eventually for the validation strategy using MRM or SRM assays, or do some multiplex assays in future. So, now the same sample you prepare in sufficient amount which could be utilized for different type of strategies. So, let us go one by one. We took the patients of meningioma, different grades of meningioma patients, especially grade 1, 2 and 3, 3 are very few. So, we had mainly grade 1 and grade 2 there are more malignant patients. We used the eye track based work strategy as well as TMT based workflows to try to compare different grades of these patients. Reliably we could get almost 3000 proteins from the quantitative comparisons, and then we also use the same samples to now look at label free quantification or data independent acquisition. Using DIA workflow, we could actually reach out now to almost more than 4000 proteins, which is after much stringent screening that we can screen so many patients, so many proteins from each patient, which now we can quantify in the label free manner. So, same patient samples, you are now trying to use either label based strategy for doing eye track based quantification, or you are using the label free quantification to compare how the controls look different from the grade 1 patient or the grade 2 patient. Similarly, we try to enrich the phosphopeptides after passing through titanium dioxide column and then use this phospho enriched fraction for the analysis, and now we try to compare all the 3 information from the label free, eye track and phosphoproteome. While each of these provides some set of unique information, but what we are also curious to see which fraction is actually showing common pattern, because that shows that these are reproducible from different independent technologies, as well as these are also showing us the trends for the phosphopeptides or the PTA modification. So, almost 208 proteins or a sizable amount of stringent proteins showed a common pattern emerging from various independent technologies. Some of those we took forward, and now we try to look at the plotting in the heat map formation, how that compares from the meningioma grade 1 and grade 2. And as you can see the green line here, this part is for the meningioma grade 2, which looks quite homogeneous, whereas the red line shows the heat map for the meningioma grade 1 patients, and this shows that there is lot of heterogeneity even in the grade 1 patient. Not all grade patients look exactly same, a set of patients look slightly different than the other set of patients. When we try to plot the PCA plots to look at the pattern based on these proteins, can we now segregate the patient population? It looks interesting that these are the 2 type of controls, dura mater and arachnoid. They are actually different from the different origin of the brain, so they look different here. The grade 1 patient look like they are forming 2 subtypes, and then grade 2 they are quite homogeneous, they look very much together. And based on these information, we are now finding some interesting protein leads, some kinases which could be taken forward. But what is most interesting from these kind of proteomics and big omics studies, you get to see large number of changes and you try to put them together and analyze based on the pathway analysis that most of the changes are perturbing which type of network and pathways. So, looking at this information, we found that integrin pathway as well as the PI3 AKT pathway were greatly affected because of the meningioma disease. So, now the logical flow was to try to investigate this problem and look at this pathway in much more detail. Many of the the network, this is what is shown here, these proteins were identified from our discovery workflows. So, our question was whether integrin and PI3 AKT pathway have some concerted effect on the meningioma pathobiology. To do this, in the collaboration from Dr. Nell's lab from Imperial College London, one of my student, she went and looked at the patient derived cell line of meningioma and then treated them with one of the inhibitor, which is ILK integrin linked kinases, to look at what is the effect of this particular inhibitor on these meningioma patient cell lines, because our aim was to look at some of the targets of the PI3 AKT pathway and whether this inhibitor could actually block or affect some of these proteins. And interestingly, it showed that this inhibitor had the effect where it could actually potentially affect the target which we have talked in the network analysis. And then after doing the real time PCR analysis, some of these targets were again confirmed that this particular inhibitor has perturbed these genes of interest. So, this study is still undergoing and currently underway to do more of the biological replicates, but the promise here is that if you can get these kind of therapies, this could be a surrogate way of adjuvant therapy where if you can't treat the patients with the surgery, can you use some inhibitors to at least try to control the tumor for some time. Additionally, the clinicians give us some sort of focus questions to address, especially based on the radiological observation, whether the skull based or supratentorial locations of the brain may have an effect at the proteome level. To do this, now if you have done the proteomics using label free, you can now use the same data and now just try to analyze that in different manners. So, in this case now we try to look at how the skull based or supratentorial brain tumors are actually getting segregated in the meningioma population. And very interestingly, it looks like they are very clear segregation of these two type of subtypes based on the radiological observation. So, looks like radiology is much more closer to the molecular signatures as compared to pathological observations. But of course, some of these we need to still take forward with more validation. But what is coming out interestingly after looking at these data and doing big data analysis from the artificial neural network, we could now see the impact of some of the positive regulators and negative regulators and how they are going to affect these type of brain tumors. So, this is where you can see that how you can start from a clinical problem of interest, identify the targets and then do big data analysis to try to get some sort of meaningful conclusion out of this information. Additionally, rather than using mass spectrometry alone, we also try to utilize the complementary technology of protein microarrays to address the question, can we identify autoantibody signatures in meningioma patients. And goal here was to use protein arrays platform, take patients clinical sample, serum sample, probe them on the chip, and if there is any antibody generated in the patient sample, they will bind on the chip which is having all the protein antigens printed. To do this work, we did collaboration from Johns Hopkins, where we have all these almost 19,000 proteins printed on the chip. Now, if we see a signal from the autoantibody which is shown in the next slide, this looks like these are the control samples, healthy individual, meningioma grade 1, meningioma grade 2 patients, and some proteins are showing very strong response, and these could be potential autoantibody biomarkers for the detection of this particular tumor. So, coming back to the various workflows which one could utilize, we have used both mass spectrometry and protein microarray based workflow. Then there is a need to do validation, and for validation you should not limit yourself to any specific technology, rather use whatever is available to you, ELISA, Western blot or microarrays, or even looking at the target based workflows to get more confidence. And many times you will see that while antibodies are not able to detect and make a lot of changes because they might have been raised for a given epitope, but your other type of complementary technologies are showing you much more higher changes, and therefore then you can gain more confidence by having multiple technologies to validate the proteins of interest. We have also tried to address specific questions from the clinicians, especially in the glioblastoma multiforme patient, looking at the location of the brain tumors, what could be the impact of the tumor location to the ventricle region, is it very close or is it far off. These patients will be surviving less, these will survive longer. It is much more aggressive tumors here, known as SVD positive or SVD negative, and can we look at the proteomic signature to try to classify these kind of tumors. In another project in collaboration from Professor Gillias lab in Tel Aviv University in Israel, we try to investigate the colorectal cancer problem, colon adenocarcinomas, and we found the panel of 9 proteins looked very reproducible across large number of patients affected from the colon cancer in the Middle East countries, and we are now trying to validate this in also Indian patient samples. Another interesting problem is medulloblastoma, the pediatric brain tumors. Again, we are trying to provide the proteomic based subtype signatures from the various type of the patients affected from either the Wnt type or the SHH type or group 3 and group 4, what could be the possible protein alteration in these type of the children, this pediatric brain tumor population. Finally, we are trying to look at variety of approaches of proteomics. Also, we are trying to use the various basic science tools of doing the transfection and understanding the effect of these mutations on this particular tumor type. We are developing various type of clinical assays in collaboration from the industrial partner of target based proteomics. So, overall I try to give you the glimpse of how to put together proteomic technologies in context of various clinical problem. We try to address 2 clinical problems of looking at the severity of infection in the case of malaria, or also looking at what could be the possible cues and subtype molecular classification of brain tumors.